All right, so it's my pleasure today to introduce Joaquin Espinosa. I don't know that Joaquin really needs an introduction here. I think everybody probably knows Joaquin very well. But for those of you who don't know his background, Joaquin actually did his BS as well as his PhD in Argentina. His PhD was done in Buenos Aires. And when he completed his PhD, I guess he decided he wanted to move to the States to do research. And he moved to the Salk Institute where he worked with Beverly Emerson. And that's where he really started his career studying p53, which I guess is what he's going to talk about today. He was obviously a very successful postdoc. He had multiple papers in journals like Molecular Cell and did exceptionally well. And so Joaquin was recruited to the University of Colorado in Boulder back in, I think, like 2004 or somewhere around that time. And spent quite a few years in Boulder working his way up the ranks. He received some really prestigious awards and grants during that time, including being an HHMI early stage investigator. So was again very successful, continued to publish very high impact papers out of his own lab, including in journals like Cell and Molecular Cell and Genes of Development, for example. So he's really been very successful with this. And in the meantime, also took on some positions in our cancer center here, running the molecular oncology program, and then also running the functional genomics shared resource. So I think through those things, many people on this campus have interacted with him quite a bit. And it was probably in part what led to his recruitment from Boulder to our campus. So he's now a full professor in the Department of Pharmacology here. But he, in addition to his cancer life, which he's going to talk about today, he is now the associate director for science at the Linda Cernick Institute, and also the director of the Human Trisome Project. So he has another life, or wears another hat, in which he works on Down syndrome. And there is some connection between the two that he's been working on. So with that, I think he's changed his title, and he's going to talk about p53 today rather than p63. Thanks, Joaquin.
Great. Thank you, Heidi. Yeah, sorry for doing this of putting a title and then talking about something else. Because of my multiple personalities, I tend to do that. So I promise you will hear about p63 in the near term. We have some really cool data on p63 and its role in promoting squamous cell carcinomas, both in the lung but also head and neck, work supported by our incipient hopefully an expert program in head and neck squamous cell carcinomas, and hopefully supported in the future by the lung sport. We'll see about that. But today, I wanted to talk about p53 because when I was putting the talk together over the weekend, I realized that this crowd, the Cancer Center, may not have heard my talk on p53 and what is it that we've been doing the last 2 years for quite a while. So I lost touch with that, that I talk to this all over the world, but I haven't presented that here in this setting. So I thought it could be beneficial because we think as a cancer center, we're in a top position, in pole position to make some serious damage in this field and have a positive impact. So for the students in the audience, very briefly, let me give you some facts about p53. Is no doubt the most commonly mutated cancer gene. This is a histogram that I put together with information on the sequencing of almost 5,000 tumors representing 21 tumor types. p53 is by far the most commonly mutated gene. This is Get Guyet's meta-analysis of the TCGA data in Nature. This is a great paper for the students, also for the faculty, where they really went home analyzing this cancer genomics data. You have Q values by tumor type, Q values. When you do a pan-cancer analysis, you see that some genes become significantly mutated only when you combine multiple cancers. Some genes are significantly mutated only when you look at specific cancer types. No matter how you do it, p53 is at the top of the chart. It's the most significantly mutated gene, both across all cancers and in many specific cancer types. So p53, the main biochemical function is a transcription factor, binds to DNA as a tetramer, and then activates transcription of genes involved in various pathways with diverse cellular functions. And out of these, somehow, this slows down tumor growth. The mutations that you see in the clinic, they all inactivate p53 activity as a transcription factor. Most of them, including the hotspot mutations, fall within the DNA binding domain of the protein and prevent either partially or completely DNA binding. And then you can have truncated mutations pretty much anywhere in the protein just past the oligomerization domain. there are very few in-frame mutations, and also very few deletions. In many cases, the mutant protein has an oncogenic gain of function. So you not only lose a tumor suppressor, but also it becomes an oncogene, and this is very, very interesting. So those POM mutations are found in about 50% of all tumors out there. If you take into account the prevalence of the various tumor types, in that guy's paper, it turns out that every other tumor in the clinic here on campus will have a mutation on p53. But on the other half, the gene may be wild type and the protein may be expressed, but it's still being shut down by other means, such as hyperactivation of the repressor p53 MDM2 or loss of the p53 activator ARF, which is an inhibitor of MDM2. So this gene circuit here, I'm going to put out this statement, and I'll wait for you to challenge me and prove me wrong, is the most commonly mutated 3-gene circuit in human cancer. You either mutate p53, you hyperactivate MDM2, or you lose ARF. So if you go to the CBio portal and look at a combination analysis of these three type of alterations, it's very easy to see that for many, many different tumor types, you reach almost 100% alteration rate. So what you're looking at here is in green, mutations in p53. In blue, deletions in ARF. And in red, amplifications of MDM2. So when you combine these three events together, for many, many tumor types, you've basically seen every tumor having at least one of the alterations. So that's my background. It's simply to say that p53 remains important despite the massive efforts in the cancer genomic field. The statement that we did 30 years ago that p53 was the most commonly mutated cancer gene remains valid today. Despite that, and the fact that there are thousands and thousands of papers on this protein, there are some big outstanding questions in the field. How exactly does p53 suppress tumor growth? You would think that we knew that by now. I'm going to show you in a minute that we are in a bit of a state of confusion about it. More specifically, what are the key p53 target genes and downstream pathways involved in tumor suppression? Is there a variety of pathways that p53 employs depending on different contexts, or is there a common pathway that is always employed consistently across all these tumor types? So those are the questions that we're trying to address in the lab. Why has this become important? Because there have been a couple high-impact papers that have really demolished long-standing views about how p53 suppresses tumor growth. For the longest time, we've been saying among ourselves that p53 can suppress tumor growth by inducing cell cycle arrest, right? Stopping cell proliferation, by inducing apoptosis. And that makes total sense. If you prevent a cancer cell from dividing or induce it to commit suicide, that should be a tumor suppressive activity. But then some high-impact papers came out showing that you can have tumor suppression in the absence of p53-mediated cell cycle arrest, apoptosis, and senescence. p53 can suppress tumor development in the complete absence of cell cycle inhibitors and pro-apoptotic effectors like p21, PUMA, and NOX. And now, in the same issue of that magazine, there is a paper with the opposite conclusion, right, saying that p53 binding cooperativity is essential for apoptosis and tumor suppression. So Savilal, the editor-in-chief of Cell Reports, calls me on the phone and says, Joaquin, I have 2 papers with opposite conclusions. Can you write a review? And I said, well, I can write a title for our review. I don't know if I can write a review. So, with Hestia, we try to elaborate on this. Is apoptosis important or not? And I'm not going to tell you the answer. I'm going to show you, data on it for you to arrive to your own conclusion. The point is that the field, it's a little lost right now. People are trying to find out, okay, what exactly is p53 doing that is so commonly mutated? Why tumors hate to have p53? But there's another side to this question, another important aspect to this question, which is that we have now p53-based therapeutics. We have drugs like Natlin and similar compounds that block the p53 repressors like MDM2 and MDM4, and they reactivate p53 when p53 is wild type. And hopefully, these drugs will elicit tumor suppression. Now, I just spent, 5 minutes telling you that p53 is the most commonly mutated gene, but, you can be optimistic and see at the glass half full, it is not mutated, it is wild type in the other half of tumors, and that is 11 million people in the world today that have tumors with a wild type version of the protein. So there are 11 million people in the world that could benefit from drugs like this. But if we don't understand how tumor suppression takes place, it's very unlikely that we will be able to deploy these drugs effectively. So not surprisingly, there's a huge amount of interest in the pharmaceutical industry on these MDM2 inhibitors. I'm listing here just some of the most prominent clinical trials for MDM2 inhibitors, different pharma, different drugs, all in phase 1, different cancer types, some completed, some recruited. So the data coming out of these clinical trials is showing what the shortcomings are going to be, which are not totally unexpected. Mostly cytostatic effects. That is, tumors stop growing, but in very few cases they actually regress and disappear. We're going to touch on that later. If you give people a p53 activating agent for a long time, some normal cell types are going to die because p53 elicits apoptosis, and that's going to lead to some cytopenias. And then, of course, like for all targeted therapeutics, if you do this for a long time, sooner or later the tumor is going to evolve around the selective pressure. You're going to end up with a tumor that is mutant for p53. But these shortcomings are not really different from the shortcomings of all biologically targeted therapies. So with this, within this framework, with this context, we set out to address two important questions in the lab. One is how exactly p53 suppresses cancer development, and then hopefully informed by this investigation, figure out ways to increase the tumor suppressive activity of p53 in response to MDM2 inhibitors. So for the first project goal, we set out to undertake the most exhaustive, detailed characterization of the p53 signaling cascade today. So what we did is to take cells with wild-type p53 and activate p53 by using an MDM2 inhibitor, shown here, NATLIN binding to that pocket in MDM2 where p53 would bind. So the drug fits into that pocket, p53 cannot bind there. Now it is set free and becomes activated. And then we employ a series of genomics analysis, what we call a multi-level omics pipeline, to follow p53 signaling across the central dogma. So we measure p53 binding to chromatin with ChIP-seq, the direct transcriptional program by this technique called global run-on deep sequencing that I'm going to explain briefly in a minute. Then we measure total impacts on the transcriptome by total RNA-seq, and then we measure translational effects by doing RNA-seq of the RNAs bound to polysomes, the ones that are actually translated into protein. So before I move forward, let me convince you, in case you didn't know or in case you came across some bad papers, that NATLIN is really, really specific. And we are really unmasking a single transactivation domain in the cell nucleus. This is an MA plot. Every dot is an RNA, highly expressed, lowly expressed, upregulated by NATLIN, downregulated by NATLIN. Red is statistically significant. You get thousands of changes in a cell with p53, but if you knock out p53, the drug is basically inert. Those small numbers is what you would get just by expected by noise. Okay? So the drug is super specific. And now let me show you some of the raw data generated by these multi-omics data sets, because then I'm going to transition into some meta-analysis. So I want to earn your trust by showing you some of these screenshots. You're looking there at the ChIP-seq data on the p21 locus. We know that p21 is a CDK inhibitor, the first p53 target gene identified about 30 years ago. We've known about these two enhancers for almost 25 years. So there you see p53 binding to the 5' and the 3' enhancer in untreated cells, and then the binding increases significantly when you treat with NATLIN. Now, GROSIC is a technology that measures the RNA that is being made de novo by RNA polymerase in the nucleus, right? So it's a true measure of synthesis. It's a true measure of transcription. So it allows you to see the entire transcript, everything from the transcription start site, well past the polynylation signal. Eventually the polymerase comes off the chromatin. You get to see both the exons, but also the introns. And you get to do a very short time point, which allows the measurement of direct effects, right? If you do a QRT-PCR or a northern blot or any other measurement of a steady state RNA at 1 hour, you can pick up activation of p21 because there is a basal amount that you need to go above. So the techniques are not sensitive, but GROSIC is very sensitive. You can go as short as an hour to look at direct effects. Then you can wait a little longer and see if that RNA that was made suffices to increase the total amount of P21 RNA in the cell, and sure enough, RNA-seq picks up accumulation of P21 RNA. Now you're looking mostly at the exons, because now you got the splicing, you got rid of the introns, you're looking at the exons. And then you can ask, is this RNA being translated? So you can purify the polysomes and do the RNA-seq, and sure enough, P21 is being translated, and it's being translated more in cells that have been treated with NATLin. And I'm going to show you in a minute at Western blot, where you're going to see this more P21 protein 12 hours after NATLin. So now we have all this information about p53 action at various stages of the central dogma. And for the first time in the history of the field, we can identify different classes of p53 regulated genes. So if we take into account all the RNAs that show increased level in the polysomes 12 hours after p53 activation, 1,200 of them, now we can ask how many of them went up in the polysomes just because there's more RNA total. So we compare that to the RNA-seq, right? So there's a fraction of genes that went up at the polysomes, but did not go up in total RNA. So those genes are translationally activated, okay? That's the easiest interpretation. There's more in the polysomes because their translation was induced, but the amount of RNA in the cell is the same. Then we can ask how many of these are true direct targets. So we cross-reference to the gross-seq data set. And we see that a very small amount, actually, are true direct targets as defined by a gross-seq at 1 hour. Okay? So when you look at the ChIP-seq data, not surprisingly, you see that the early direct targets, nearly 80% of them, have a p53 binding event within 25 kb of the promoter. This is what you would expect for direct targets, p53 binding near the promoter. And that is significantly more than you would see at all genes, on the genome average. Now, we notice that in this group, those that did not score in gross-seq but went up in RNA-seq, we still have significantly more than on the genome average. So what happens here is that p53 triggers an early wave of transcription that we pick up by gross-seq, but then it may trigger a secondary or later wave of transcription that we cannot pick up by gross-seq. But now we can inform ourselves with the ChIP-seq data to try to identify the direct targets in this group. So what we did is to look at the spatial distribution of p53 binding in the true direct targets, and that told us that p53 binding to the direct targets is significantly higher than the genome average, only up to 2.5 kb from the promoter. If you go beyond 2.5 kb from the promoter, you can tell a direct target from an indirect target from a false positive. This is the first time in the history of the gene expression field where we can actually have an informed decision about how to use ChIP-seq and RNA-seq data. Because you gather your favorite paper, people do ChIP-seq, RNA-seq, and they pick up an arbitrary window from the promoter. They say 10 kb, 20 kb, 5 kb, whatever, your choice is as good as mine. Well, now we can have an informed decision based on the gross-seq data and the ChIP-seq data on the gross-seq targets that for p53, the right window to take into account is 2.5. It's not a perfect window. We may miss some direct targets that have a more distant enhancers, but if we were to expand our window, we're going to get a lot of false positives, right? So when we take this window, we now recover a group of direct targets, but they are induced late. So these genes have a p53 binding site within 2.5 kb, just like the direct targets. They go up at the RNA, and they get translated in the polysome. So we should be looking at these genes as well. So here are some examples of the signals that we're getting from the various omics data sets. The translational activated genes do not go up in gross-seq, do not go up in total RNA, but they go up at the polysomes. The indirect genes do not go up in gross-seq, do not have a nearby p53 binding site, otherwise they would have been in this group, but they do go up in total and polysomal. And then we have the true direct target genes like p21 that go up in every one of the assays, okay? So now we can do this for the repressed genes. And this is very interesting and somewhat controversial in the field. Basically, we don't see any direct repression. There's people that believe that p53 is a transcriptional repressor, which, actually we don't. We don't understand how this protein with two very potent transactivation domains can also have a very potent transrepression domain somewhere in the polypeptide. We see only one gene that is reducing synthesis at the gross-seq level. And then we see 11 genes that at the RNA-seq are going down and happen to have a p53 binding site within 2.5 kb. But that's what you would expect by chance. Because I told you that if you look across the genome, 13% of genes have a p53 binding site near the promoter. So this is not statistically significant. Now, sure enough, there is a lot of gene repression, both in total RNA and also translational repression. It is real, but we believe it's all indirect, and we have made this argument in other papers and have shown the mechanisms of repression. It's basically action of microRNAs and action of P21 and other CDK inhibitors. So for the first time, again, we get to see the p53 signaling cascade with this degree of detail. And now we can do this in multiple cell types, right? For the first time in the history of this very crowded field, we can ask, how does p53 signaling vary from one context to the next, right? So we took 3 different cell types, different origins, vastly different mutational spectra. Take my word for it. These come from different people. They had different mutations, and they had different responses to the drug. So the cell type that I showed you so far, ACT116 from colon cancer, does not die in response to the MDM2 inhibitor, remains viable, no signs of apoptosis. This osteosarcoma cell line we chose because undergoes apoptosis in response to the drug. You can see there activation of p53, p21, but also activation of caspase-3. And then this breast carcinoma cell line has an intermediate phenotype. So here we have 3 cell types. They're very different, have different responses. We can ask, what is the degree of conservation and diversity in the p53 network? So we did the same exercise, the same meta-analysis of the various omics data sets. And there are some similar features. There's always a small direct program and a small late direct program, then massive expansion of p53 signaling at the indirect level, and then an intermediate number of genes that are regulated at the polysomes. And then repression is largely indirect, it's large in numbers, and there are also some differences in the amount of genes that are regulated in every cell type. So for the first time in the history of the p53 field, we can ask, okay, how different or how similar is p53 function across different cellular contexts in tumors derived from different people? So I'm going to show you a set of overlap Venn diagrams preserving the size proportionality, relative to number of changes. When you look at the direct program from the get-go, only 25% of direct early target genes are conserved. So as early as we can measure, there are already significant differences between these cell types, right? Then when you look at the late direct response, there are even more differences. You have massive expansion at the indirect level, even more differences, less conservation. When you get to the polysomes, every cell type is doing something different, right? Massive diversity, massive cell type-specific action of p53, massive cell type-specific action of this biologically targeted therapy, an MDM2 inhibitor. When we look at the repressed genes, again, very little or non-direct repression. There is more overlap here in the indirect repression. And if you look at these genes that are loaded with genes that are specific, transcripts that are specific to the S phase, histones, DNA polymerases, MCN complex, this makes sense. The cells are arresting first. Some of them are going to die. And as they arrest, they shut down transcription of S phase transcripts. That's what you see in there. Again, the translational response is purely cell type specific. Every cell type is doing something different. Now, this to me as a biochemist, and you may be fooled by looking at all this genomics data, thinking that I was trained as a genomicist. I was not. As a biochemist, it makes total sense. I hit a biochemical reaction here, and as the sound expands throughout different rooms, you get propagation of diversity. And small differences in the initiating event become propagated into big differences in downstream signaling cascades, right? A note about quantitative effects. It's good to look at all these numbers when you set up a threshold and a p-value. But what about the intensity, the size effect? The direct targets, although few in number, and you can see this box and whisker plots, they are narrower because there are fewer genes in the direct program, tend to be induced more strongly than the larger number of indirect and translational regulated genes, okay? So they are direct, they go up first, when you measure the polysome, you see a greater fold change. So the way we interpret this is that p53 signaling ripples along the central dogma, right? Look at the picture of a ripple. You drop a drop, and you get, the first wave is very deep where the drop fell, right? But it's small. That is the direct program. Then you have a second ripple that is shallower, but it's bigger. And then you have a third ripple, right, that is even shallower, of intermediate size. So that's what p53 is doing through the central dogma. Direct, indirect, translational, strong effects, mid-size effect, more shallow effects. So now that we have these various categories of p53 target genes, we can start asking regulatory questions. We can ask, what defines whether a direct target is early or late? Could it be that a direct p53 target gene is early in one cell type, but late in the other cell type because of some regulatory differences? So we can ask that now. There are many differences. The early targets bind more p53. These are cumulative distribution plots, and you can see that the early direct genes are shift to the right, meaning they bind more p53 as a group, as a collective. They bind more p53 than the late direct targets. In turn, those bind more p53 than just a random collection of p53 binding sites across the genome, not associated with regulation, okay? So they bind more p53. The enhancers of these early direct targets are pre-activated, they are prime. You may have heard about enhancer-derived RNAs. Enhancers are the sites of the DNA where transcription factor binds, but it turns out we learned recently that they also get transcribed. They don't make any protein, there's no protein coding information in enhancers, but they make RNA, and you can pick up those RNA by grow-seq. So when you look at the eRNAs of the enhancers associated with the early direct targets, even before you activate p53, look at the DMSO lines, the faint lines, there is a lot more eRNA transcription than at the late direct targets or at the non-direct. So these enhancers associated with the genes that fire first are already kind of active, and they tend to bind more p53. They may be binding a small amount of basal p53 even before we treat with an MDM2 inhibitor. Another feature that distinguishes the early from the late direct targets is RNA polymerase action. We can look at the RNA polymerase across the entire body of the gene in early and late direct genes. And as you can see, the early direct targets, even in the DMSO, in the untreated cells, they already have more RNA polymerase sitting at the promoter ready to fire into the body of the gene. This is called RNA polymerase pausing. The polymerase binds to the promoter, but doesn't go. It is waiting for a stimulatory signal, like p53, for example. So there's more polymerase waiting for activation signals at the early direct targets. Now, these early direct targets, because they go up first, when you look at the polysomes, you see more induction than to the direct late, either polysomes or total mRNA. So they go up first. If you stop at 12 hours, and probably true at any time point that you would do this experiment, you get a greater fold induction for the genes that go first, okay? So now, if you assume, like some people believe, that p53 uses a consistent pathway of tumor suppression, that it always does the same trick regardless of context, so then that pathway should be in the core program. Forget about all these differences that I should show you among cell types. If there is a core program, the tumor suppressive pathway should be in that program. So we set out to identify this program that is conserved across cell type. And that is what is shown in this upset plot. So what we're looking at here is basically a cross-reference of the various data sets, the early direct, late direct, and indirect genes for all three cell types. And you come up with a group of 103 genes that are activated directly by p53 in all three cell types that we looked at. For example, 31 genes are called early direct in all three cell types. Now, notice how many genes that were called indirect in one cell type were called direct in another cell type. Those are the genes that have the very distal enhancers. Remember that we use a 2.5 kb window, which is not perfect? So sure enough, there are genes that have an enhancer 5 kb, 10 kb. Now we call them, because they were gross-seq targets in one cell type, but indirects in the other. So they should be reclassified as direct target genes. So what are they? What is these 103 genes? is the who is who of the p53 signaling cascade, right? It's all the major effector pathways, and multiple genes per pathway. Many genes involved in cell cycle arrest, many genes involved in apoptosis, many genes involved in differentiation, DNA repair, metabolism, so on and so forth. So p53, regardless of context, always activates a mixed bag of signals, a multifunctional core program. Now, the cells may end up doing different things. I showed you one of the cell types, HCT116 cells, obeys this program and remains arrested for days after drug treatment. Now, SGSA cells start doing this, but then they transition to this pathway and undergo apoptosis. But p53 told all the cell types to do everything, regardless of context. And then somehow the regulatory landscapes of each cell type define the cellular outcome, okay? So this is good news, because, why is it good news? Because it means that every tumor that we get our hands on and treat with NATLI, we're gonna have activation of many cell death genes. And we have spent a huge amount of effort in the lab trying to understand why the cells are not dying. And you can read some of these papers, okay? But every time we use the drug, we have increased apoptotic signaling. That is good news, even though in most cases the cells do not undergo apoptosis. Now, this core program is very interesting. It's driven by high affinity enhancers that are bound in every cell type. So this is the ChIP-seq data, one cell type versus the other one. You can see the various pairwise comparisons up here. And then we see that all high affinity binding is conserved, right? These are the high affinity enhancers. They are gray, meaning common for both cell lines. The cell type specific binding in the various colors is low affinity, right? So if you have a high affinity enhancer, p53 will bind in a breast cancer cell, in an osteosarcoma, in a colorectal. It won't care. It will bind it everywhere, right? Now, low affinity enhancers, yeah, those may be regulated in a cell type specific manner. With this background, the enhancers associated with the core program tend to be high affinity enhancers. And that is also shown here. The core genes in every cell type bind more p53 than those that are direct only in that cell type, right? So the core program has the best enhancers. Again, makes sense that that's why they are core, because they have the strongest p53 response elements. This is unusual in the field of gene expression, right? I don't know how familiar you are with transcription factor action and chromatin and nucleosome enhancers. If you read that literature, transcription factors are supposed to act in a cell type specific manner depending on the chromatin landscape, right? Now, p53, by and large, has more binding events that are common than there are cell type specific, say, for example, in this pairwise comparison, HCT versus MCF7. Now, you take your favorite transcription factor in the serum response network, that's not what you see. Most binding is cell type specific. Now, if you look for the p53, for the DNA sequence underlying these peaks, you basically get the same sequence with small variations, right? But if you do that here, you get two different motifs, right? Maybe there's a companion transcription factor, maybe there is some other determinant of binding, you end up binding different DNA sequences. Same is true for a large group of transcription factors for which we have data on the two different cell types. You know, I could do this all day. It is very unusual to find a transcription factor that has such a degree of conservation in binding. Now, this shouldn't have been surprising to me, especially because the first experiment that I did when I moved to the United States, I was in California, and I moved to California to surf. And I had long hair, you know, and I went to the lab in shorts and flip-flops. The first experiment that I did, when I still had hair, was this DNase 1 footprint analysis. For students in the room, this involves radioactive. Did you know that we used to do things with radioactive? Is that still, are there still radioactive licenses in this campus? So what you're looking at here is a piece of DNA with a 5' or the 3' site enhancer in the P21 promoter, incubated in vitro with pure p53 increasing amounts, nanomolar. And you can see the protection of p53 on that sequence. DNase 1 cannot cut, cannot give you a radioactive piece of DNA of that size. You can see that footprinting, although it's slightly shifted, even when you wrap up that DNA around nucleosomes. p53 is very special among transcription factors in the sense that can see its binding sites, even when they are wrapped around nucleosomes, a sort of pioneer factor, okay? So that may be why it can see high affinity binding sites in all kinds of chromatin landscapes, in a breast cancer cell, in an osteosarcoma cell, to activate the core genes. So now that we know what the core genes are, and we have a big idea of why they are core, we can ask, okay, which of them mediate tumor suppression? Right? It's 103 of them, give or take a few. Which ones matter? So to address this question, we employ genetic screening strategies in human cells. So if you're in this campus, thanks to the functional genomics facility, chances are you have heard about these shRNA screens before, so I'm going to explain this briefly. You take cells, transduce a large library of shRNAs, every cell gets a different shRNA, and then you can split that Petri dish. One is untreated, or vehicle. The other one is treated with nothing. p53 is activated. In this case, we're using SGSA cells. They arrest and then die. And we do the experiment at 48 hours, so we leave some cells alive in the Petri dish, about 20% of them or so. And then we let those cells recover, and then we hit them again with nothing. And after 2 rounds of nothing treatment, we harvest these shRNAs in the surviving cells. So if we knock down something in the p53 network that is required to kill or do cell cycle arrest, that shRNA now should be more abundant. The cell that has that shRNA has a selective advantage. That shRNA should increase in abundance. So we did that with SGSA cells for a couple of reasons. First of all, SGSA cells have MDM2 amplification. It's very unlikely that they have any other inactivating mutations in the p53 network. They make a lot of MDM2. If you inject them under the skin of a mouse and let a tumor growth, you treat with nothing, the tumor disappears. Okay? So p53 is intact as a tumor suppressor, at least from a therapeutic point of view. So when we did the experiment, the only gene that showed consistent enrichment of shRNAs targeting that gene was p53 itself. So we did this with a p53 target library. So we collected all the genes that we call as direct targets by our various omics analysis, spike a little bit of p53 shRNAs as a positive control. Our positive control shRNAs took over the population of shRNAs, within 2 rounds of treatment. No other p53 target gene could confer in this setup a selective advantage to an MDM2 inhibitor. So we said, well, maybe none of the p53 target genes is very strong, but there is a factor outside of the network that may be very strong as a tumor suppressor working with p53 when you activate p53. So we repeated the screen on a genome-wide library, and we got basically the same result. The only gene in the genome that is mediating p53-dependent tumor suppression is p53. So p53 needs itself to do what it does. And you will say, well, it doesn't need anybody else. No, that's not the right interpretation. It needs a lot of helpers where each one helps only a little bit. So there's no second in command in this network. There's no second, there's no lieutenant that gets 50% of the activity. No. p53 as a transcription factor activates 103 genes, and chances are each one of them gets 0.5% of the activity, or 1% of the activity. So we said, well, maybe we're not doing this right. Maybe we should do more rounds of nothing. We should create more evolutionary space, extend the timeline, see if we can pick up the second base. It may be, contribute only to 5%, 10% of the activity, but it has to be a second best. So we did that. More rounds of nothing. All we achieved was to select for cells with mutations in p53 in the Petri dish. Within 2 rounds of nothing treatment, you can already see the p53 mutants start taking over the culture. And because they are oncogenic, they have a proliferation advantage. Within 4 rounds of nothing, 90% of the population has hotspot mutations in p53. So the experiment is impossible. We cannot find the second best in the network. There isn't such a thing. The only way of shutting down this network is getting rid of p53 itself. And that is why it's the most commonly mutated cancer gene. Okay? So sure enough, the cells become totally resistant to the drug within 4 rounds of treatment. This is very scary, right? As we're going to the clinic and we want to give people this drug, within 4 rounds of treatment, you get resistance, and 90% of the cells have a mutation in p53. And then, I'm not going to, overwhelm you with a lot of genomics analysis that we did, trying to find a p53 target gene that was significantly mutated in cancer in a mutually exclusive fashion with p53, right? So we have those 5,000 tumors that TCGA has sequenced. We could not find it. I'm just showing you this snapshot just to make my point. That's the mutation rate for p53. This is the mutation rate for the core targets. Take my word for it. When we look at every one of these, none of them is significantly mutated in a consistent mutually exclusive fashion with p53. A few of them are in some very specific context, maybe in one tumor type or the other, but none of them can really bypass the need for p53 mutation. So to summarize this part of the talk, if you go to that list of the core target genes, you will find genes that p53 activates involving every hallmark of cancer. p53 can shut down proliferation, induce apoptosis, try to induce differentiation, block angiogenesis. Every one of these hallmarks is represented within the p53 network. So if you inactivate any one branch of the p53 network, you don't get a tumor because you still have, all the other tumor suppressive mechanisms in place. What you need to do is get rid of the node in the network to shut down all those, tumor suppressive pathways at once. And that is that p53 mutation itself, or MDM2 amplification, or RFLOS, what gives you a very strong selective advantage in the evolutionary setting of an incipient tumor. So the p53 signaling cascade is massively diverse across cell types, lots of differences, but within those differences, there is a core program that is activated by p53 consistently. And the activity of p53 as a tumor suppressor is what is spread very thinly, very evenly across many of these genes. Some may matter more than others, but we can't pick up which one it is that matters the most, at least not with the tools available to us. Therefore, inactivation of p53 itself provides the most powerful selective advantage during tumor evolution. So now that we know that, how do we employ this knowledge? How do we make p53 a stronger tumor suppression in the clinic? Okay? Let me remind you what the problem is here. You get cells with wild-type p53 and treat them with NATLIN. p53 becomes activated. You activate 103 genes that I told you, plus many more. Now, in most cases, you're going to get cell cycle arrest. The cells are not going to die despite the fact that many apoptotic genes were induced. In some select cases, like this cell line here, you're going to get activation of caspase and apoptosis. That's the tumor you want to have. You want to pop a pill and 16 hours later, you want the tumor to die. But that's very rare. How do we make that event more frequent? Okay? Now, the good news is that the resistant cells have an intact apoptotic machinery. These are resistant cells. They don't die to NATLIN. But if you give them a nasty genotoxic agent that makes your hair fall off and wipes out your gut and your immune system, yeah, those cells will die. Okay? Because this is super pleiotropic and does all kinds of nasty things. And that cell death is largely p53 dependent. But these drugs, of course, activate many other things. The point of this slide is that the cells have a machinery for suicide. It's just that the clean drug is not fully engaging that machinery. So knowing this, we set out to do synthetic lethal with NATLIN screens. And this data is a few years old, so I'm going to go quickly through it. Many of you may have heard this. It's basically a screen to look for genes that can convert, that when knocked down, can convert the arrest into cell death, right? The survival genes that are holding away the apoptotic genes, right? The survival genes. If we can shut them down, now the drug should kill. So we did that with a standard synthetic lethal approach. Cells will arrest if you treat them with NATLIN. If you hit a survival gene, they're going to die. When the student comes in the weekend and dumps the media into the sink, the cells go into the sink, the shRNA that you care about went into the sink, into the sewage. How do you find it? You sequence the shRNAs here and look for the delta, okay? You look for the shRNAs that are still in the vehicle-controlled plate but are gone from the drug-treated plate. So Kelly Sullivan in the lab did that, and sure enough, he found many shRNAs that you can count with a deep sequencer, very obviously in the vehicle-treated cells, gone from the naturally treated plate, okay? Many. So I'm going to speed up here a little bit. It turns out Kelly found 2 druggable nodes that are loaded with synthetic lethal genes, the ATM signaling module and the MET signaling module. The negative numbers represent the mean drop-off in abundance of shRNA in the naturally treated cells. The 2 big circles are druggable protein kinases. So ATM It's a protein kinase that exists both in the nucleus and the cytosol. When you block ATM, nothing happens. When you activate p53 with natrine, you get p53 activation but no cell death. When you combine the two clean drugs, you get p53 activation and cell death as much as the dirty drug 5-fluorouracil that makes your hair fall off. Okay? You treat tumor spheroids with a drug alone, you get cytostatic effects here, nothing much happens here, but then combined, you wipe out the tumor spheroids and you get great synergistic index. Same for MET inhibition with crizotinib or with other drugs, or by knockdown. If you block MET with crizotinib, nothing happens. But if you block MET with crizotinib and activate p53, now you get a conversion from arrest to cell death as much as with the dirty drug, and now you wipe out the tumor spheroids. Okay? This is very spectacular. You can kill these cancer cells without radiation, without a cocktail of DNA damaging agents. You block one E3 ligase, MDM2, you block one kinase, ATM or MET, boom, they're gone. Okay, so we can do this in a smart way, knowing exactly what we're doing without having to use cancer-causing agents. Did I say that right? Yes, we treat cancer patients with cancer-causing agents. Did you all know that? We should stop doing that, because in 100 years from now, our lineage is gonna hate us. So check this out. When we block these kinases, the core program is intact. The core program doesn't change, okay? CDK inhibitors, BH3-only proteins, activated with or without active ATM, with and without active MET. What happens now is that we enable p53 to activate more apoptotic signaling in the so-called death receptor extrinsic apoptotic pathway, activation of caspase A, the extrinsic caspase, cleavage of beat into truncated beat in both cases. And you need these two to get killing. When Kelly knocks down or knocks out these two, you no longer get synergistic killing, okay? So this TB, biochemically speaking, is the same than PUMA. It's a BH3-only protein that will block many BCL2 survival factors and engage the intrinsic apoptotic machinery. So from this slide, if you are still awake, you can predict two new combinatorial strategies. Natrine should work better if you engage dead receptors with a dead ligand, and it also should work better if you feed cells a drug that looks like a BH3 protein, a BH3 mimetic. And sure enough, that's true. We had published that before, right? So these results kind of came back and confirmed things that we knew already, that combining BH3 mimetics with natrine will induce cell death. There are many, many papers now in the literature showing that tray ligands plus natrine will kill cancer cells. Okay, so I shall show you two more combinations. Let me show you three more. If you know your ATM biology, you would probably be aware that ATM is a DNA damage activated protein kinase that is supposed to activate p53 by phosphorylating both p53 and MDM2. And that should contribute to apoptosis. But when we don't use DNA damage, when we go after MDM2 with natrine, ATM has this other opposing role of preventing apoptosis. It's a synthetic lethal gene. So what is this about? This is not the canonical function of ATM. It's not the nuclear function. Its function in mitophagy. What Kelly found is that when you block ATM with a drug, within hours you accumulate all mitochondria. Accumulation of all mitochondria correlates with increased levels of mitochondrial reactive oxygen species. And those ROS then tip cells over into apoptosis. And sure enough, the combination of an ATM inhibitor and an MDM2 inhibitor does not work if you use an antioxidant. You can prevent the synergy by blocking ROS production. So then Kelly looked into the mechanism further and said, well, what if I block mitophagy or macroautophagy by other means? What if I block autophagy with chloroquine? That works too, right? So when you block ATM, you get increased autophagy as seen by these markers here, and even more when you do the combination treatment. So when you treat with chloroquine, when you block autophagy on its own, nothing much happens, but when you combine that with p53 activation, now you get synergistic killing. That's my fourth combination for you. When Kelly looked at the pathways downstream of MET action, and MET is a receptor tyrosine kinase that engages many downstream pathways, he found that the key pathway for synthetic lethality was this transcription factor, STAT3. What he found is that you can reduce STAT3 activity just by half, by toning down the amount of STAT3 phosphorylation with this drug is static. STAT3 total levels are the same. Phosphorylation goes down 50%. That's all you need now to kill these cells. This is a spectacular result because for the longest time, we thought that transcription factors were not good drug targets, right? Somebody came out with the idea that kinases were good drug targets. Now, let me tell you, all 2,000 kinases come from one ancestral gene, okay? They're all the same, in my view. You can put them in a single phylogenetic tree. They all do the same. Take ATP and put a phosphate somewhere. You know, somehow we invested zillions of dollars on trying to find specific inhibitors of a family of proteins that is very similar. Transcription factors are not like that. They come in all kinds of flavors. They come in all kinds of surfaces. Now, sure enough, they don't have a catalytic activity that you can drug. But that doesn't mean they're not druggable. Here, we are activating one transcription factor and bringing the activity of another one by half. That's all it takes to kill a cancer cell. So transcription factors are good drug targets. We just need better chemical biology. So that was my fifth combination. Let me show you the sixth. This phosphatase WIP1 is another negative feedback loop within the p63 network. It's a p53 target gene. It's a phosphatase that will dephosphorylate a lot of things to keep p53 activity low, okay? And recently, this is a small molecule inhibitor of WIP1. When you use that inhibitor into cells, p53 works better as a transcription factor. Now you have increased transactivation of many apoptotic genes, also many cell cycle arrest genes. Now p53 works better. Target genes are induced to a higher level. Does that mean the cells will die now? Yes, right? This is Senex's work in multiple cell types. You get very low levels of apoptosis in these cell types when you use natrium alone. But if you combine now with a GSK compound that blocks WIP1, you have increased apoptosis. So that's another way of making p53 better, by shutting down two of its repressors, MDM2 and WIP1. Let me show you another potential strategy. In collaboration with Haida, they had this beautiful data showing that 6-1, an oncogene, in a number of settings, shuts down p53. Here's an experiment of natrium treatment where you treat these cells with natrium. You know, they arrest, some of them die. But if you overexpress 6-1, you confer resistance to the drug. You look at gene expression data for 6-1 across hundreds of tumors, and those that express more 6-1 tend to be natrium resistant. So you may say, well, maybe we shut down 6-1, natrium, will work better. Can we shut down 6-1? Not 6-1 per se, but 6-1 has a cofactor, which is also a phosphatase called AYA2. And Heidi and Rui Xiao have developed now inhibitors for AYA2. So it may be that we have now a potential way of combining AYA2 with natrium for synergistic killing. We haven't done the experiment, we should probably do it soon. So my last slide. Since 2009 or so, or 2010, when we set out to start looking for these combinatorial strategies with Kelly and others and the creation of the functional genomics facility, we had come up with many, many potential strategies, right? That will make natrium kill cancer cells while preserving at least non-transformed fibroblasts and other normal cell types. I told you about trade ligands, BH3 mimetics, ATM inhibitors, many inhibitors, STAT3 inhibitors, autophagy inhibitors, WIP1 inhibitors, maybe messing with 6-1 levels or activity. Many, many strategies. Now, the bottleneck now is to take this to the clinic. So the MDM2 inhibitors are not FDA approved for anything yet. There's a lot of ongoing clinical trials, but they are not approved. That prevents us from doing a lot of things in terms of combinatorial strategies. But we are engaging, various people in this campus across the cancer center to start moving these things closer to the clinic. With Anthony and with Victor, we're working in liposarcomas. This is a great tumor type for MDM2 inhibitors because 95% of them have MDM2 amplification and wild-type p53. And Victor and Anthony are already giving MDM2 inhibitors to some of these patients. Now there is an ATM inhibitor that has good in vivo properties that pharma wants to put into people, and we're working with Wells and Todd trying to get the preclinical research done. I was super happy to hear from Mandali that she's now in charge, basically, if I understand correctly, of a clinical trial by Amgen to combine natrium with BH3 mimetics. I show you the paper. We proposed this in 2008. It's been 9 years ago that we've been telling the world, if you increase the BH3 load, p53 is going to kill better. So that clinical trial is recruiting. We have great experts in autophagy here, and we have some of them who are putting autophagy inhibitors into people. We need to do the necessary work now to combine those with MDM2 inhibitors. And many, many other people. I hope I don't offend anyone by not showing the picture. So in conclusion, p53 consistently activates a multifunctional program. In most scenarios, the outcome is arrest. However, with synthetic lethal screens, we have uncovered many signaling cascades that protect from the killing effects, leading to the design of rational synthetic lethal combinations. Credits where they are due. I hope I'm not forgetting anyone. These are the 4 postdocs, I believe, that did all the work that I showed today: Senek, Matthew, Kelly, and Anna. I think of the PRs that are still in the lab, Amanda was involved in what I showed you today. Of course, we couldn't do this without the amazing support of the Functional Genomics Facility, many, many collaborators and funding sources. And with that, I'll be happy to take your questions and criticisms. Thank you. Heidi. Correct, you got that right. So p53 is perfectly designed to keep you tumor-free until a lion eats you in the African savannah. They are very rare in the wild. Okay, pediatric tumors are very rare. They seemingly don't impose that much of a selective pressure, to increase the tumor suppressive capacity of this network or to have a backup. What happens when you crank up too much p53? There's a deleterious aspect to having too good of tumor suppression, which is premature aging and senescence. You start, you are on an easy trigger, right? I should say there are mechanisms to make a better tumor suppression network with p53, and that is on big mammals, right? So you may be aware of the Pettos paradox, where if you look at a little mouse and you look at an elephant, you would say, well, elephants should have a lot more tumors than mice. That's not true, right, in the wild. And that's the Pettos paradox. Why mammals that have way more cells don't have way more tumors? Well, in the case of elephants, they have like 12 copies of p53, right, which are retrogenes, are truncated proteins that spring-load the network. So you have a lot of p53 poised for rapid action upon activating stimuli. So there are ways through evolution, to have a stronger, better p53 tumor suppressive network. The network that we have is perfectly fine until we start getting, vaccinated, living longer and whatever, living past our reproductive age. That's when the problem starts. Puma is there, yeah. Yeah. Yeah, Puma was there, P21 is there. So I think these papers were right. Yeah, you can get rid, of one gene here or there, and p53 has a lot of other options, yeah, a lot of other mechanisms to induce tumor suppression. Here and here. For different transcription factors? I think it would be variable. I think p53 tends to act more distally than many, many transcription factors. When you do a meta gene or a meta enhancer for p53, there's a bit of a distribution outside of the promoter. Many factors, like for example, MYC, SP1, they're very close to the start site, within the first couple hundred base pairs. So I'm open to the possibility that, that window would be different for different transcription factors. Yes. We haven't. We have, look at some phosphorylation marks in 15, this and that. We haven't done an exhaustive, characterization of the PTMs and whether that is a contributing factor. of any kind. Some people believe that these decorative post-translational modifications can impact the choice of p53 target genes. I'm less convinced of that, but it remains possible. Yes? No, MDM2 is amplified with extra chromosomal copies. If you do a fish of this nuclei, these big blobs of MDM2 DNA, that doesn't go away. When you put the drug, all that MDM2 no longer works. So the only way of becoming resistant to the drug is mutating p53. I used to ask my cancer biology students in Boulder, how do you become resistant? Do you mutate MDM2 or p53? And many will say, well, you mutate MDM2 where the drug will bind. Well, if the drug can't bind, p53 can't bind either. And so then you have, you don't get resistance that way. The effector of the drug is p53, not MDM2. Some people have tried this, and it depends on the stress. For example, people have thought that natrium actually would be protective from mitotic poisons, right? Like taxanes and that kind of thing. Because by inducing cell cycle arrest first, right? If you do a temporal sequence of treatment, if you put natrium, most cells will arrest. Then you put taxanes, you don't have undergoing mitosis to poison mitosis. So in that case, natrium actually would counteract the action of those stressors. But in other cases, it would synergize. Or the genotoxic agent will be dominant. When you mix natrium and 5-FU, you get cell death when you put them together, right? And I think we've done back in the day actually a sequential addition of drugs, putting natrium and then 5-FU, cells die. So you can, people have done quite a bit of this, of mixing natrium with standard chemotherapeutics, and again, depending on the poison that you're talking about, you may get synergism or you may actually get protection. Great. Thank you, everyone.